Hello everybody, welcome to my channel by Shruti. I am from the SNDT Women's University of Mumbai. Today I am going to introduce to my topic which is instrumentation of HPLC. HPLC means high performance liquid chromatography in which high performance stands for high resolution power and liquid stands for the mobile phase in which liquid form is used in the analysis. So, as per the examiner point of view, we usually draw the block diagrams to explain this instrumentation part because it's the simplest and easiest way out there. But like me, have you ever thought that how does this analytical instrumentation operates inside or how does a sample component separates out? So this point will be explained to you by me today regarding the HPLC instrumentation with the help of some virtual diagrams for the better understanding. So let's start. First, we'll be coming to the instrumentation part. Okay. The principle of HPLC is based on classic column chromatography. HPLC is an analytical technique used to separate, identify, and quantify various components in the analyte mixture. As well as the other form of chromatography, there is a mobile phase which passes the degasser. It is then further forced by a pump to pass through the column system. Do you know what happens in the column? First is the equilibrium, second, binding, third, washing, fourth, and the last step is the elution of the separated component. Stationary phase is a column which is placed as a separate compartment where temperature can be controlled using the oven. The sample can be automatically injected in the HPLC system by using autosampler or by using the sample injection system. In autosampler, the sample is manually inserted by using a syringe. The last segment of this instrumentation is a detector part, which is attached to the system at the end, which helps us to detect the specific analyte after the separation in the column. This all overview comes under the hardware part of the system. There is one more part known as the software, which has a data system. After separation of each component, the eluted component is shown in the form of graphical representation. Next, we'll move on to the working system of HPLC. This is the instrumentation diagram shown. As we discussed in the previous slide, there are two solvents A and B. One is the volatile, other one is the non-volatile. Below the solvent reservoir, there are two pumping stations individually for A and B. In HPLC system, the purging pump and degasser both are connected to the pumping solvent system A and B, where they are used to remove the dissolved gases from the solvent. The purging pump is filled with the inert gas, which helps us to remove the uncertainty in the solvent in the form of impurities. The pump drives each solvent A and B into the mixing chamber, where mixing takes place under the high pressure. Sample introduction can be accomplished in various ways. The simplest method is to use the injection valve. Currently, auto-samplers are built in the HPLC system, which helps us to preserve the sample and analysis takes place with very low quantity and more efficiently. HPLC analysis completely uses isocratic elution method where mobile phase remains constant throughout the procedure, whereas in gradient elution method, the mobile phase changes its composition at the separation. Once the sample is injected into the auto-sampler, the switching valve switches its position and sample then passes through the separator column where separation takes place efficiently and the eluted component is then passed to the detector and detector use the UV visible method to detect it and interpret the data. Now we will see how does the sample injection system works or operates. Have a look on the virtual image. In the load position at high pressure, the mixture of solvent flows through the column directly. Then, the loop is loaded with atmospheric pressure. The sample through the syringe is inlet at the needle port. Excess of sample is drained out as a waste form. After loading the sample, the valve switches its position to the inject position. This is the inject position, where the flow of solvent is delivered by a pump, forcing the sample to move ahead through the column efficiently. The sample is moved ahead. Then, the valve returned back to the load position. Mobile phase moves the sample through the column. This procedure repeats until sufficient amount of sample is being separated out through the column. This is the first load position, then comes the inject position. Moving ahead, type of HPLC on the basis of column used. The separation is based on differential partition of the sample component between the mobile and the stationary phase used. The component which have more affinity towards the mobile phase will travel faster and get eluted first, and the one which have less affinity towards the mobile phase will travel slower. This is the diagram shown where the yellow components is the polar one and the red one is the non-polar one. In comparison to the red one, yellow have the more affinity towards the mobile phase, that's why it's traveling faster in comparison to the red analytes. The separation can be carried out on which type of HPLC is used. First will be the normal phase HPLC, where stationary phase is a polar and the mobile phase is a non-polar molecule. Just see the diagram. Over here, polar stationary phase used is a silica gel. In this case, hydrophobic analyte will travel faster with this mobile phase. Second is the reverse phase HPLC, where stationary phase is a non-polar and mobile phase is a polar molecule. Non-polar stationary phase used is silica C18. In this case, hydrophilic analyte will travel faster with this mobile phase. Once the component is eluted out of the column, it is then passes towards the detector for the analysis. Detectors. In HPLC, the UV visible type of detectors are the best one to be used for the instrumentation part, as it shows the absorption spectra in that ultraviolet and visible region by measuring the absorbance of analyte using Beers-Lambert's law. A deuterium discharge lamp acts as a UV source and a tungsten lamp acts as a visible source. A mirror is used to show both the lamp to the diffraction grating and it disperses accordingly to the wavelength. When measurement is to be performed with specific wavelength, the angle of grating can be adjusted according to it. So the light of the wave must shine on the flow cell while doing the analysis. Half mirror is placed between the diffraction grating and the flow cell, where the specific wavelength is split into two parts, one to fall on the sample side, other one to fall on the reference side. The eluted component from the column enters the flow cell, where bonding and non-bonding electronic compound can absorb this energy in the form of light and gets excited to the higher energy level. The final data is interpreted in the software attached to the system of HPLC. The analysis is observed in the graphic form where x-axis as a timeline and y-axis shows the absorption measured. Graphical representation. The data which is observed at the software part of the HPLC instrumentation is represented in the graphical form where x-axis is at a timeline and y-axis has the absorbance. The number of peak is equal to the number of analyte observed. So this data can be used as a quantitative analysis where determination of concentration is done. There are two methods. One is determination of the peak height, other one is determination of the area under the peak. Coming to next, this data also can be used for the qualitative analysis where retention time can be helpful for us. Retention time means time taken by the solute to pass through the column. It helps us to determine the identity of the compound. There are various factors affecting the retention time such as glass flow rate, temperature difference, column degradation, etc. The irregularities in the graph with multiple peaks makes it difficult for the identification. So, use of reference standard can eliminate those errors and help us for the better observation. Some of the important formulas are also given which helps us to solve this problem related to the chromatographic technique. First one will be the retention factor. Second one is the selectivity. Third will be the efficiency. Fourth will be the resolution power. Thanks for watching this video. If you do like, please subscribe my channel and press the like button. In case of any queries or any questions related to this video, you can let me know in the comment box below. Till then, stay tuned